Yes, I'd like to sort of highlight one area which is usually overlooked when it comes to sort of proteomic experiment. A lot of focus is generally placed on the mass spectrometer because it's very expensive, and that's where the results are generated. But it's important to remember that your results are never going to be better than your sample. If your sample are low quality, regardless of the amount of money you spend on the mass spectrometer, the results are still going to be low quality. So, and I represent a company called Denator, and we manufacture the instrument, the stabilizer, for stabilizing the molecular composition in the sample. So it's alive. This is sort of a central theme of what we do. And it's, in this case, is of course the sample. It's very important to remember that once you take the sample, the cells in the sample are still very much alive. And they don't know that sort of they've been removed and they're doomed. They're trying to survive. Because what happens when you remove a sample, I'm primarily speaking about a solid sample, like a tumor sample or a biopsy sample or something like that. So first of all, you have a loss of blood flow. And it's the blood that transports oxygen and nutrients onto the tissue. So very quickly, you will have loss of oxygen, hypoxic situation. That will cause the cell to switch to an anaerobic metabolism. So instead of using oxygen as the sort of end product, as in generating ATP and energy, they will start to sort of generate lactic acid instead. And lactic acid accumulation will lead to a lower pH. And this is also leads to, since this is much poorer using oxygen and doing glycolysis, and the whole lactic and the whole respiratory cycle is much more efficient when it comes to generating energy in ATP. So very quickly we have a very low ATP environment in the cells. So all of these are powerful signals that the cell will sense and try to adapt to. And it does so by changing phosphorylations, digestion proteins, and so on. And of course, in a longer scale, it will also change levels of transcription and translation and so on. But in the very first phases it will start by the primarily phosphorylations, and also lipids will start to degrade. So this is one area from the moment the sample is taken, or really from the moment that if it's an animal, the moment the animal dies, that's when the blood flow stops. That's really when the clock starts ticking and these changes start to occur. So that's one sort of danger zone until you either freeze the sample or heat stabilize it, as we are doing. The second sort of danger zone is when you start to extract your sample. Somewhere down the line, you generally homogenize your sample and extract it. And even with inhibitors, even with 8-molar urea, even with high levels of organic solvents, there are enzymatic activity, and they will change the sample composition. There's some examples of that later in. So that's another area where you can have changes. And so what we want to do, we want to abolish, remove enzymatic activity, because the majority of these changes are due to enzymatic activity. So by removing the enzymatic activity, preferably as soon as possible after sample collection, we stop this cascade, or at least the changes brought on by this cascade, and you also stop these changes that happens during after homogenization and extraction. So first, there are some examples of how does it look? How do I detect changes, post-sampling changes in the sample? And then I want to present the stabilizer as such. And then there's a number of examples where we do comparison between using the stabilizer and using state-of-the-art inhibitors and rapid freezing. So what we see here is a mass spectrometry. It's an LC on this axis, and then some mass spectrometry analysis on this. So LC-MS analysis, and we're detecting peptides. So we've used, this is mice brain extract. We've used a molecular weight cutoff filter, and that will remove everything which is larger than 10 kilodaltons. So we're only looking at small peptides. And the idea here is that that's where we find the, in this case, neuropeptides. So this image, the left image, has been done from heat-stabilized samples. And we see that the majority of the peaks here have been identified as being sort of no neuropeptides that you expect to find there. A non-stabilized sample with only 10 minutes post-mortem. And this 10 minutes here doesn't really mean much, because the majority of what we see here is loads and loads of peptides. But when we go in and identify this, the majority of these are degradation fragments of albumin and other sort of housekeeping large, high abundance proteins. That was for peptides. So peptides are one of the class of molecules that we see change rapidly post-sampling. The other one is phosphorylations, protein phosphorylations. So what we have here is a reverse phase protein array experiment, similar to a Western blot experiment. It's a time course experiment, so we have mice brain. We remove them as fast as possible and then freeze them rapidly. We kill the mice, we wait 10 minutes, then we remove the brain. Or we kill the mice, wait 30 minutes at room temperature, then collect the brain and freeze it. And then we do this and analyze, extract them in, there was a 6 molar urea buffer, and we look at how the levels of these 11 common phosphorylation change. And all of these, some of them decrease quite rapidly, some not so much. The majority of decrease seems to happen during the first time period compared to the second time period. This has probably to do with the disappearance of the ATP that happens quite rapidly in brain tissue. However, what's interesting to note, this is a study that we did. This is another study which is published that we have nothing to do with. They're using a different tissue. So this is uterus tissue. Some of the phosphorylations are similar. It's a slightly different time scale, slightly longer. Their 0 is not quite at 0. There's actually a little longer here. But anyway, what's interesting here is in this system, the majority of the phosphorylations actually increase in the initial period, and then they decrease. So we don't really know why that is, and this is a completely different tissue. And so it's important to realize that phosphorylations, some phosphorylations will go up, some phosphorylations will go down post-sampling, and this may change depending on tissue. And also there will be a lot of phosphorylations that we'll see later on are stable. It doesn't change at all, but quite a few will. So again, unless you know how your phosphorylations that you're interested in study, how they change, it's important to do time course studies to understand, are my phosphorylations that I'm interested in affected by post-sampling changes? This is another example. This is more specific. We're looking in this is brain tissue, protein called CREB, a specific phosphorylation. So this is immunohistochemistry. A whole brain is either stabilized in the instrument and then placed into formalin, or directly placed into formalin. And then we're visualizing this phosphorylation on CREB. And this is the one that goes, and then we cut the sections so that we have the full gradient. So this is a coronal section that sort of goes into this is cerebellum, so the hindbrain. So, and we see here a clear gradient. This is the outside. So the formalin penetrates from here and in this way. And we see that where the formalin penetrates rapidly, in the outer layer, we have a good signal. However, as it goes in, we start to have a fainter signal, and in here, there's basically no signal at all. Because formalin penetration but depending on tissue, is roughly between 1 and 2 millimeters per hour. So these are generally takes quite a long time to stabilize, and that's what we see here. During that time, there is still enzymatic activity which can change the phosphorylations. Here are some so this is where it's been stabilized, where the enzymes activity is removed. We see activity, we see this phosphorylation sort of all the way from the surface and inwards. And if we stabilize it and leave it in room temperature 24 hours and then put it into formalin, we'll still see very similar staining. However, if we have an ordinary brain, only 2 hours at room temperature, and then stabilize it, we don't see anything. There's just a tiny, tiny bit on the very edge here where oxygen penetrates from the atmosphere. So phosphorylation. So another group of molecules that change rapidly are free fatty acids. So lipids are generally stored as triglycerols. And so each triglycerol has 3 fatty acids. And in an effort to recover some little energy, these fatty acids are released in the post-sampling case. So in this case, again, time series experiment. In this case, it's the homogenate. And this is mainly to illustrate that in the homogenate, there is an active enzymatic process ongoing that releases these free fatty acids. And again, we see some are not specifically affected, just a very modest increase, whereas some increase very rapidly. Again, emphasizing, unless you know how your specific lipids or molecules are changing, you don't know. They may be changing a little, or they may be changing by several 100%. So it's important to investigate these parameters. So that's the few examples of sort of what you can see if you do time course experiments. So this is the problem that we're addressing. And to do this, we stabilize through thermal denaturation. So we use heat, a heat-based technology. And in the next slide, I'll show you exactly how it's done and what happens on the molecular level. So, so we have the instrument itself. There is a cartridge. This is the cartridge for solid tissue. There's a cartridge for liquid samples. There's also a cartridge for DBS, dried blood spots. So this system then can either be used very close to the sample source and having the instrument sort of directly and introducing it directly into the instrument. It's also possible to use with frozen samples, so that you freeze the sample rapidly after excision, transfer it frozen, and when you have it sort of with the instrument, it's brought frozen into the instrument and directly from a frozen state heated. So what happens in the tissue itself or in the sample itself is that, so enzymes are proteins and they have a 3D fold. And this 3D fold is very detrimental. It's very important for the activity. So, so what we do here, we take the sample, we apply heat. So we add energy, so we heat the sample to roughly 90 degrees centigrade, and that causes the proteins and the enzymes to unfold into a linear, non-active state. And then the sample is taken out of the instrument, it cools down, and through the sort of hydrophobic collapse, we will get another 3D fold, which is generally a denatured, non-active state. And this will further, as this is done sort of in the tissue, there's lots and lots of proteins in there, we will get an aggregate coagulate state of the proteins locking in this inactive new form of the, that's how it works. So it's a very basic, works for more or less all proteins. The one exception that we know of are RNases. RNA degrading enzymes are very short, and they're held together by two disulfide bonds. So they denature, but the disulfide bonds stay intact, and when they cool down, it will fold back into a native configuration. That's the one sort of class of exception that we have found so far. But the majority of protein-affecting enzymes seems to find sort of a new denatured stable state after they've been heated. So this is a short little movie of how a sample is stabilized in the instrument. So that's the card, and sample goes in the card. It's sealed, and as you see, the upper part here is sort of a dome-shaped. So the first thing that happens when it's in the instrument is that air is evacuated. That's to make sure that the sample is the highest point on the card. As it goes in, the height is measured, and based on the height and whether or not the sample is fresh or frozen, the time it takes to stabilize will be calculated. The sample is heated, it returns out, and it's done. And then the sample is ready for either if you want to extract it right away, or if you want to store it for a few hours, it can be fine at room temperature. If you want to store it for a longer time, we recommend that you freeze it, preferably at -80. So for a small, like a 2-3 millimeter sample, like a biopsy, like a small piece of sample, it's roughly 20-25 seconds. If you have a full-size mice brain, 6 something like that millimeter, it's roughly 1 minute. The maximum size height is 7 millimeters. So it's a small sample. You cannot put a whole human in there or anything. It's a fair amount of sample. So what happens to enzymatic activity? What we have here is an experiment to visualize phosphatase activity. So phosphatases removes phosphorylations. And we're using a substrate called PNPP that when the phospho is removed, it turns yellow. So what you do is you do homogenate of your sample. In this case, it's mice brain. In PBS, you add the substrate, you incubate for roughly half an hour at 37 degrees, and then you measure the yellow color. And that's what we see here. So without inhibitors, we see lots of yellow color, lots of phosphatase activity. This is what we expect to find in PBS in a sample. If we add inhibitors, this is the phosphostop inhibitors from Roche, we see there's a marked reduction, but nonetheless, roughly 50% of the activity remains in the sample. If we compare that to the activity level that we detect in the heat-stabilized samples, we see that that level is really on the background level. So very low to background level activity detected after heat stabilizing of these samples. So the other part of the equation is the kinases. The kinases adds phosphorylations to proteins. And so this is an array experiment. You have an array with 144 peptides. Each peptide contains a phosphorylation site, a tyrosine phosphorylation site. You incubate your extracted samples with in this array, and if there are kinases in this mixture, they will phosphorylate these motifs. And then you use a tyrosine-sensitive specific antibody to detect the level of phosphorylations on these peptides. And so here are the 18 most abundant, most phosphorylated motifs. And again, we see that in the snap frozen, we see quite high levels. And in the heat stabilized, there is just 3 that shows any type of activity. And there is another 120 or so peptides. And if all these are summed together, and we do a percentage comparison, we find that 99.6% of the kinase activity has been abolished in the heat-stabilized samples. So those were the examples of sort of what happens to the enzymatic activity. That will. So now let's get over more into the technology and the comparison between using heat stabilization and using standard workflows. So it's an upstream technology, and that means that it comes very early, close to the sample source. So and afterwards, you can do most ordinary proteomic techniques from Western blot, 2D gels, over through LC-MS-based technologies, phospho shotgun, and so on. We've primarily focused on peptides and protein phosphorylations. That's where we see that those are the classes of molecules that change most abundant there, where the change is most abundant following post-sending. There are examples of proteins which are also degraded, but generally proteins are not degraded as rapidly as phosphorylations and peptides. We've also started to move into, there are some examples of that, into the lipids, small molecules like pharmaceuticals and so on. Here's an example of two peptides, dynorphin A, dynorphin B, and they are enzymatically converted in the body to this shorter version, lev-enkephalin arginine. And when we measure these and using a RIA, radioimmunoassay system, we find that the two sort of parent peptide, the dynorphin A and B, we see higher levels in the heat stabilized, that's the D, compared to the snap frozen. However, when we look at the sort of cleavage product, we see much higher levels in the snap frozen sample. It's important that it's illustrated. These samples, they're taken, they're rapidly frozen. From a frozen state, they're dropped into boiling 1 molar acetic acid, boiled for 5 minutes, and then they're extracted, and then the extract is again heated to 95 degrees for 1 minute. That's the sort of snap frozen. That's the standard workflow that they use for these samples. The only difference with the heat stabilized are that directly after sampling, they're introduced into the stabilizer, snap frozen, and then they're extracted and boiled and so on the same way as the other. So despite these efforts, boiling in this 1 molar acetic acid, very acidic solution, we still have this sort of continued degradation. Another peptide example, this time this is a technology called multi-imaging. You have a thin section of a mice brain, in this case. So what we have here, that's the cerebellum, the hindbrain. You have the cortex up here. And we're looking at a peptide called PEP19. It's rather large, 6,715 is the mass. And so in the full-length peptide, we see the distribution pattern in this tissue is relatively equivalent, slightly higher in the stabilized tissue, but nonetheless, more or less very similar. What's interesting is when we go down and look at the two other masses, which have been identified as being degradation fragments from this full-length peptide, we see that they are only found in the snap frozen where we have enzymatic activity. And they have roughly equivalent distribution as you would expect from a degradation fragment. So moving on from the peptides to protein phosphorylations. This is a phospho shotgun, so a bottom-up approach. The sample is digested with trypsin, extracted 8 molar urea, digested with trypsin, and then titanium dioxide are used to enrich phosphorylated peptides. In the heat-stabilized samples that's been into the stabilizer, we find roughly 4,400. So this is 3 replicates. If we wait, we kill the animal, wait 5 minutes, then heat stabilized, we see a bit of a drop, 4,200 roughly. If we wait 20 minutes and then heat stabilize, we see a further drop to roughly 4,000. And we compare that to the snap frozen. So the snap frozen in this case, the brain is removed, put into liquid nitrogen, homogenized in liquid nitrogen, and then 8 molar urea with inhibitors is added to the sample, and then you do the extraction and so on. And we see despite the measures, 8 molar urea, keeping it cool during extraction and so on, we still see a further reduction to roughly 3,700. So this again is since it's sort of impossible to know how many phosphorylations are there in the tissue originally, a tool that we generally use is this time course studies. By following what happens in the time course post-sampling, you get sort of an idea of how does my molecules of interest change. So on average, the majority seem to be that there's a loss of phosphorylations. So anything which is less is probably, with time, it loses with time, it sort of changes from its original composition, and this change is a removal of phosphorylations. So this sort of tells us that this sample with fewer phosphorylation is longer from the original in vivo situation compared to what we have over here, when we see that it's sort of the original, maybe one could have detected 5,000 phosphorylation, for example. That's impossible to know. So this is a pre-study. This was unfortunately never published. He then went on to use the heat stabilizer together with other techniques to study phosphorylations in roughly 14 different rat organs, and discovered roughly 36,000 phosphorylations in these 14 tissues. That data is published. This is data from another experiment that we did in collaboration with a company in the US called Cell Signaling Technologies. They primarily produce antibodies. And what we see here on this axis is the ratio between the intensity for a peptide in the heat stabilized sample divided by the intensity in the snap frozen sample. Again, snap frozen rapidly after excision, homogenized frozen, extracted in 8 molar urea with inhibitors. That's the standard workflow that we're comparing with. And so anything here, the orange bars, will have had at least 50% higher intensity in the heat stabilized samples compared to the snap frozen samples. And there's also a few that decreases. So in this case, they would have had higher level of phosphorylations in the snap frozen samples. So these would have sort of added phosphorylations during the extraction and postmortem. These would have lost phosphorylations during the extraction. So roughly 1/3 of the phosphorylations, the tyrosine phosphorylations that we detected in this study, have changes at least 50% from what we think compared between these two experiments. So it's also important to note that roughly 2/3 did not change. So a lot of phosphorylation doesn't change. So that's so if you're lucky and you're only looking at 1 or 2 phosphorylations, you may be looking at a stable phosphorylation. That's good for you. But you don't know until you've done the experiment. The time course experiment will tell you whether or not your phosphorylation is stable or not. So if we dive down to some specific proteins, we see here, this is the breast cancer anti-estrogen resistance protein. It has some sort of connection to breast cancer. We find 3 phosphorylations in the heat stabilized samples, whereas only 1 and only very little in the snap frozen sample. Another example is tau. Again, happens to be there's 3 phosphorylations in the heat stabilized samples, 2 phosphorylations in the snap frozen sample, and again we see here that the levels are highly statistically significant. That was the examples for phosphorylations. The other example is what I showed initially, the free fatty acids. So just to the time course experiment in the homogenate, some increase rapidly, some not so much. When we do the direct comparison between the heat stabilized sample and the snap frozen sample, so these samples are extracted in acidified methanol, so 100% or 99.9% methanol, 0.1% TFA. So acidic methanol, very organic, very denaturing solution. Nonetheless, we see large differences. In some tissue, again, a difference in tissues. So the 14 here, we see almost no difference in the brain, large difference in the liver. I don't know why, but that's just the way it is. And some tissues behave differently than others. So the majority of the free fatty acids show much higher levels in the snap frozen, and we're pretty positive that this is due to this ongoing enzymatic change in the sample. So these were examples from solid samples. It's also possible to do the more liquid samples, like cell cultures, plasma, blood, CSF, something like that. So we have a card, we call it Maintainor Liquid. And so the tissue had a dome shaped to be able to sort of fit around a physical sample. The liquid card consists of two membranes, and when you add a drop of liquid in here, the liquid will be between, will sort of stand between the plastic, the Teflon membranes. And regardless of volume, from roughly 50 microliters up to 1.5 milliliters, it will have the same height. And the height is really what determines the time it takes to heat something. So I also have one example. We just recently released these, but we have one example where we sort of stabilized a liquid sample. In this case, it's a yeast extract. So it's yeast. It's not an extract. It's live yeast cells. So they're grown, they're harvested and washed, and then they're suspended in PBS, and then they're heat stabilized. And then we did the extraction in 8 molar urea and separate them using 2D gels. And in this case, we focused on these. There's 7 very prominent spots in the standard proteomics buffer extraction, with sort of direct extraction, that are pretty absent in the heat stabilized samples. And when these were identified, it turned out that all 7 spots were from a protein called enolase. Enolase is presumed to be the theoretical sequence. It's a pretty large protein, so pretty high up in the gel. That's another indicative. If you start finding proteins, identifying proteins down here, which have a theoretical molar weight up here, then you pretty much know that what you've identified is a fragment. And it's also noteworthy that sort of the background, there's sort of a hazy background to this gel that we don't find here, because all proteins doesn't degrade as nicely as this one. But maybe say that 1 to 5% is degraded. That doesn't change the pattern very much, but it does show up in a sort of a background smear that we don't really see here, which is sort of gives a cleaner. That was the example for the liquid card. We also have another liquid sort of card, dried blood spots. And the dried blood spots, that's a technology where you put drops of blood on a filter paper, and then you let this filter paper dry. And once it's dry, it's stable, and you can send it through ordinary post, and you can store it at room temperature. And this is generally a very good system for collecting samples, like in rural areas. And if you have to have lots of sample and don't want to invest a lot of money in freezers and so on, which can be very, very expensive. So, however, the problem here is that from the moment that you put a drop of blood on the filter paper card until it's dry, depending on humidity, roughly 1 to 2 hours. And during this time, you have a liquid sample at room temperature with enzymatic activity that can degrade your sample. So what we do is that put the drops on there, put it in the instrument, heat and activate the enzymes, take it out, let it dry, and then you go downstream analyze. We've analyzed, together with a group in Thailand, a number of pharmaceutical compounds. They want to do DMPK studies, how quickly does a molecule, a pharmaceutical that you take, how quickly is it digested in the body. Very many pharmaceutical compounds are differently digested between different people. And then it's important to know if you're a high digester, you need to have a higher dose in order to have the same sort of benefit, same efficacy on the drug. And if you're, because otherwise you will have a very low level of active drug in your system, and you may not have the, and vice versa. If you're a very low digester, you may get very high, you get toxic levels, when that is not good either. So there's a number of examples in the paper. Here are some of the examples. So this drug, esmolol, it's given as a prodrug, and esterases in the blood converts this into the active form. And this is another form that is, this is the given, this is what it's converted to. Again, a time course study to see what happens post-sampling. In this case, what happens after it's placed on the filter paper. So the blood is collected, it's spiked with the, so we add these two compounds as spiking compounds, pure synthesized compounds, into the blood. And then we either stabilize, put the drop on, stabilize directly, wait 1 minute, 2 minutes, 5 minutes, 10 minutes. And we see here, the blue, that's the mother, the one we spiked with. The level stays roughly similar. The degradation fragment, the active molecule, slowly increases with time. And in the non-stabilized case, where we let it just dry at room temperature, the way this system is supposed to work, we see quite high levels of the degradation product. If we delay the drying, we put it in a plastic bag for 1 hour before we remove it and let it dry, we see that this sort of conversion continues. So the conversion continues as long as it's wet and as long as there is enzymatic activity. And when we remove the enzymatic activity, we see very low levels of the degradation product. This example is even more striking. Same type of experiment, and here it really shows if you wait 1 minute, 2 minutes, 5 minutes, 10 minutes, the mother compound is rapidly degraded. And here, already at the time 0, more than half of the compound that we added have been converted to the degradation fragment. And when we look at the non-stabilized, the standard way of working with this, we see that almost all have been converted into this active form. That was the examples. Some words of using the heat stabilizer in a clinical setting. The majority of work that's been done is done in the university setting, laboratory animals primarily. It is being used in some hospital setting for clinical research. It's not in routine use, but people that are interested in changing the way treatments are being done, in a few cases, primarily involving biopsy sampling. So in this case, the patient in the operating room is surrounded by a sterile zone. And we have the instrument sort of on the border between the non-sterile zone and the card itself sort of goes in, and the surgeon either drops the piece of tissue in the card or hands the the forceps with the sample, and it's placed into the card. That's generally how it's used. The stabilizer can be integrated sort of in the workflow. The samples generally emanate from some sort of operating room. They generally go through a pathology, an analysis lab, and sometimes part of the samples end up in a biobank. So the samples can the stabilizer can either be integrated right where the samples are taken, and then sort of stabilized with as short post-sampling time as possible. It's also possible to integrate it at the pathology lab, to freeze the samples, have them frozen, and then when they arrive at the pathology lab, you can do the stabilization. We've also done studies taking sort of fresh samples that have between 30 and 45 minutes post-operation time, and even with that time, we see an improvement in the number of phosphorylations that we detected. So it's also possible to have it here, or as it arrives at the biobank, or also here as the biobank sends out the samples to customers. And there's a number of pluses and negatives depending on having it in these various. So apart from stabilizing the molecular content of the sample, there is some other potential benefits with using the system. So a couple of years ago, a researcher with the US Army, Lisa Casares, she approached us at a trade show and said, I think I have a killer application for your instrument. I want to test that. And what she wanted to do was to use the heat stabilized system to inactivate bacteria, because she's working in a BSL-3, BSL-4 high containment laboratories with very scary diseases. And in order to get samples out from there, she had to put the samples into formalin for 21 days. And after that, it was practically impossible to do any sort of molecular characterization of these samples because everything was so tightly fixed in the sample. She's gone on to test the system, and the first publication is out. So in this publication, she shows that she infects laboratory animals with either it's a virus, a model virus strain, or a bacterium. And then she does a sort of a dilution, takes a sample, homogenizes, and dilutes to see the number of live pathogens that she has in the various tissues. And for the bacterial case, primarily she finds it in the lung and to some extent in the spleen, in the standard non-treated animal, in the non-stabilized animal. In the stabilized samples, she cannot find any live bacteria of this species. And it's the same thing for this encephalitis, so it's a brain virus. And she's found it in the brain, not in the spleen and kidney. So that's the way it's supposed to be. However, after heat stabilization, she's not able to detect any live bacteria. So I'm just so she's now sort of expanding this and to see which different type of bacteria and viruses this can be useful for. I generally see this as a way to reduce the unknown loads. If you have if you have samples from, say, a human sample, and you don't really know what type of pathogens this patient may or may not have, this can be a way, in addition to stabilizing the biomolecules, it can also reduce the level of pathogens in the sample. We don't guarantee anything with this application, so you sort of have to verify this through your regulatory, but we definitely think this is an interesting side benefit, and it's being tested right now with the CDC for tuberculosis samples, and so far it looks good. So we'll see what happens with this application. Another sort of side benefit which we see every now and then, quite often in many studies, is an increase in reproducibility. So what we see here is a comparison of the intensity. This is a shotgun experiment, and we're comparing the intensity of the peptide in sample 1 versus sample 2. And if it's more in sample 1, it will end up on this side of 0. And if it's more in sample 2, it ends up on this. And we see that for the heat-stabilized samples, the samples are much more similar compared to the snap frozen. Let me see here. A better way of showing this is through using a PCA plot. So principal component analysis, where all the variability in the sample is sort of plotted onto 2 or more, in this case, 2 principal components. So every spot becomes, every sample becomes one spot. So in this case, with the sample, with the dots with the same color, that's one sample group. They all have the same treatment. When they cluster closely together, that means that they are more similar compared to the groups over here, which are more spread apart and are more different. And so these are various extractions of peptides using different extraction buffers, all urea-based, and after that there's some various incubations at generally 37 degrees. This was an experiment we did to show that there are enzymatic activity in 8 molar urea buffers and 2 molar urea buffers and so on, but it also shows quite elegantly how the 2 heat-stabilized groups cluster very closely together as well as within their groups, whereas all the snap frozen, the regularly without the heat stabilization, they're both more spread apart, the groups, and also within the group you have a sort of wider spacing, indicating that they are more dissimilar, more different. Yeah, that was the last of the examples, and I just want to finish off by really pointing to, unless it's unclear to anyone, that we think that it's very, very important that rapid changes are happening post-sampling, and it's key to doing high-quality biological studies to standardize sampling and stabilize the molecules. And we think that the stabilizer system can be one component of this standardization. I just want to add with a little self-promoting. This is a small article I did a few years ago. It's called The 11 Golden Rules of Working with Proteomics Samples. It's freely available on the web, and there's 11 sort of points, goes through the sampling all the way from the collection to the data analysis, and sort of points out what are important to consider through the entire experimental phase. So just Google the golden rules of proteomics, and you'll find it.